welcome to the digestive system. We're looking at the last part of our four-part series, our very last video. In this video, we're going to look at the large intestines as well as the minerals, vitamins, carbohydrates, proteins, and lipids, and how they are digested within the system. So let's take a good look at our large intestine. What is the large intestine? It's about 1.5 meters long, or 5 feet long, and it receives approximately 500 milliliters of indigestible residue per day. Now the residue is about 150 milliliters of feces by absorbing water and other salts. And it's eliminated by defecation. So look at the size of it, as I mentioned earlier. It's about 5 feet long, 2.5 inches in diameter. And that's just looking at it at a cadaver. And it has 4 major regions. And look at those 4 major regions. It has the cecum, which you see here. Okay, we have the cecum, the colon, then the rectum, and the anal canal. So remember, the large intestine has 4 regions: the cecum, the colon, the rectum, and the anal canal. Now let's take a look at the cecum here. The cecum is like a little pouch in this lower right abdominal quadrant, and it's inferior to our ileocecal valve, or the ileocecal sphincter, as you see right here within this particular diagram. Now, attached to the lower end of the cecum is the appendix, and it's a blind tube, and it's approximately 2 to 7 centimeters long. It is a significant source of immune cells. Okay, so the colon is that part of the large intestine which you'll find between the ileocecal junction and this rectum. So this entire thing here, structure here, this is considered to be the colon, and that is divided into 3 regions. That will be your ascending, our ascending, our transverse, and our descending. Those are the 3 regions of the colon. Now we look at the ascending colon. It begins at this ileocecal valve or sphincter, and then it pushes up the right side of the abdominal cavity. Then it marks this little 90-degree turn at this section here called the right colic, or the right hepatic flexor, and it's near the right lobe of the liver. So the liver will sit right in here. Then we have our transverse colon, and it passes horizontally across the upper abdominal cavity, and it makes a 90-degree turn downward at the left colic, or the left splenic flexor near the spleen. Alright, so this is our right colic, or right hepatic, and this is our left colic, or left splenic. And these are called flexors. Alright, now here it becomes the descending colon. And this is going to pass down the left side of the abdominal cavity, all the way till we get to the cecum colon, as well as to the rectum. Now, right here, the pelvic cavity is narrower than the abdominal cavity. So at the pelvic inlet, the colon turns inward, or it turns medially and downwardly, form this S-shape portion called the cecum portion. Now, sometimes you'll have an instrument that's used called the sigmoid scope, and is used to look at the sigmoid portion of the colon. Also in the pelvic cavity, that large intestine, it continues as the rectum, which is about 15 centimeters in length. So here we have a large or an enlarged view of the rectum. Now the rectum has three folds called transverse rectal folds or rectal valves. Now those valves, they allow the passing of gas while retaining the feces. Now the final 3 centimeters of the large intestine is the anal canal. Is this anal canal? Now the anal canal, it passes through the levator ani muscle, and then it's going to terminate at the anus, which is right here. So this is the anal canal, and here is our anus. Now the mucosa of the anus, it forms these longitudinal ridges. So we see right here, we have these ridges right in here. And those ridges are called the anal columns. Okay? And then between those depressions, now those depressions that you see between those columns are called anal sinuses. So here are our anal columns, and those deep impressions here are called our anal sinuses. Okay. Now the passage of feces, it causes the sinuses to exclude or extrude, extra mucus to lubricate the anal canal during defecation. Now the large hemorrhoidal veins, which you see right here, it's not labeled in the diagram, but these little blue circular structures or circular things here on the diagram, those are our large hemorrhoidal veins. They form these superficial plexus in the anal column and around the orifice. Now they lack valves and are subject to distension and venous pooling. Now hemorrhoids are permanently distended veins that protrude into the anal canal, or they form bulges that are external to the anus. So you have some of them that are external, that will extend outward of the anus area. Okay. Now the muscularis externa of the colon is unusual in that although it completely encircles the colon, its longitudinal fibers are concentrated in 3 thickened ribbon-like strips. Now each strip, we call here, each strip is called the taenia coli. Now the muscle tone contracts the colon lengthwise and causes its wall to bulge, forming pouches called haustra, which you see right in here. These are called haustra. Now in the rectum, in the anal canal, however, the longitudinal muscle, it forms a continuous sheet called the haustra, which you see right here in the diagram. Now the anus, right here, the anus is like the urethra. It regulates by 2 sphincters. We have an internal sphincter, and we have our external sphincter. The internal anal sphincter is composed of skeletal muscle of the pelvic diaphragm. No, the internal anal sphincter is composed of smooth muscle of the muscularis externa. But the external anal sphincter is composed of that skeletal muscle of the pelvic area, of the pelvic diaphragm. So here's our external anal sphincter, which is voluntary, and then we have our internal anal sphincter, which is involuntary. Basically, we cannot control that, but we can control the external anal sphincter. So of course you may see that on a quiz. Might be a true or false question, maybe multiple choice, might be true or false. Might be the internal anal sphincter is a voluntary muscle and made up of and composed of skeletal muscle of the pelvic diaphragm. So make sure you pay attention. If I'm stressing it, you need to pay attention to it, or write extra notes here on the side of your PowerPoint. Okay. Alright, so the ascending and the descending colon are considered to be retroperitoneal, whereas the transverse, remember the transverse is one that goes across, the transverse colon and the cecum and the sigmoid colon are covered with serosa, and they're anchored to the posterior abdominal wall of the mesocolon. Now the serosa of these three regions often has epiploic or omental epindays, I'm sorry, and those are like flat, fatty pouches of peritoneum, and of course they do not have a function that is known to us. So here's a diagram, a gross diagram of the large intestines. Here we have our cecum, we have our ascending, transverse, descending, we have our sigmoid, the rectum, and we have our anal canal. And of course, here is that region where the ascendant takes that 90-degree turn horizontally. This area, this section here is called a right colic flexure. And this area here is the left colic flexure, where the transverse colon makes a 90-degree turn downward to start forming that descending colon. Here's our sigmoid colon, which makes that S-shape part of the colon, and here's our rectum area and our anal canal. And you see that you can visualize the external canal, the external anal sphincter wrapped around. You see it's circling the anal, or the anus, or the anal part of the anal canal. And of course you see our appendix area, and here's our ileum from the small intestines. So that's why they have the ileocecal valve, talking about the two areas. The ile deals with the ileum, and the cecal deals with the cecum. So that's why we have the ileocecal valve. And of course we talked about that in the last video, where the ileocecal valve basically regulates the substance, the feces, or the chyme coming into the cecum, into that large intestines. Okay. Alright, and here's just a closer view of the anal canal. Here we have the rectum, we have the rectal valve, here we have our anal canal, our anus, and here's a closer view of the anal columns that we talked about, and those depressions that you find between each column are the one is called the anal sinuses. Here's the internal sphincter. The internal sphincter, what do you say, is composed of smooth muscle of the muscularis externa, and then we have the external sphincter here, which is composed of the skeletal muscle of the pelvic diaphragm. And which one did I say is voluntary? That's right, the external anal canal, external anal sphincter, sorry, is voluntary, and our internal anal sphincter is involuntary. You can't control this one, but you can control the external. Alright, so in terms of the microscopic anatomy, the mucosa of the large intestine has a simple columnar epithelium in all regions except the lower half of the anal canal, where it has keratinized stratified squamous epithelium. Now there are no circular folds or villi in the large intestine, but they are intestinal crisps. Now these crisps are deeper than in the small intestine and have a greater density of goblet cells for increased mucus secretion. Now the lamina propria and the submucosa have an abundance of lymphatic tissue, and they provide protection from the large population of bacteria in the large intestines. Now the large intestines, of course, they're going to harbor a number of bacteria. About 800 species of bacteria collectively, and they're called bacteria flora. Now the relation with the bacteria is mutually beneficial. They provide us with additional nutrients that we cannot extract elsewhere, and some also synthesize the B vitamins and the vitamin B and K vitamins are synthesized, which are absorbed by the colon. Now, bacteria produces intestinal gas, which is a less desirable effect. Now, the large intestine, it contains about 7 to 10 liters of gas, expelling about 500 milliliters per day as flatus, and reabsorbing the rest. So we hear flatulence, flatus. We're dealing with intestinal gas. Now, most is swallowed air, but the flora adds to it. Now, cramping up results from undigested nutrients that are passed into the colon and furnish an abnormal substance that leads to increased gas absorption. Now, flatus is composed of nitrogen, carbohydrates. Let's see some of this page. Yep, is composed of flatus is composed of nitrogen, carbon dioxide, hydrogen, methane, hydrogen sulfide, and two amines, and they're called indole and skytol. Now, indole, skytol, and hydrogen sulfide, they produce the odor of the flatus and the feces. So remember those three: the indole, the skytol, and the hydrogen sulfide. They're going to produce the odor of the flatus and the feces. Let's see if it's on this page here. Here we go. I kind of jumped ahead of myself. Here we go. The hydrogen sulfide, indole, and skytol, they produce that odor for the feces and the flatus, which is the gas. Alright, so the large intestine, it takes about 12 to 24 hours to reduce the residue of the meal to feces. So I have here 36 to 48. It is 12 to 24 hours. So the intestines, let me see. Alright, so the intestines, it does not chemically change the residue, but it reabsorbs water and electrolytes from it. Now, feces are 75% water and 25% solids. Now, the solids are 30% bacteria, 30% undigested dietary fiber, 10 to 20% fat, and smaller amounts of protein, sludge cells, salts, mucus, and other secretions. Okay. Alright, so now the fat that is not from the diet, but from broken down epithelial cells, and of course from bacteria. So the fat that we have is not from the diet, but from the broken down epithelial cells and the bacteria. Now the most common type of colonic motility is segmentation, and that's called haustral contractions, and they occur about every 30 minutes. Now, distension of the ostium with feces. Remember we talked about the ostiums are those thick bulges. All right. So the distension meaning they're going to stretch out. But these feces, it stimulates it to contract, which churns and mixes the residue, promotes absorption, and passes the residue distally. Now, strong contractions called mass movements over 1 to 3 times a day, and they last about 15 minutes and move residue for several centimeters at a time. Now, mass movements are often triggered by gastrocolic and duodenocolic flexures, in which filling of the stomach and the duodenum stimulates the motility of the colon. Just give me 1 second. Okay. All right. I keep jumping ahead of myself with these slides. So we're here on slide 12. We just talked about the gastrocolic and duodenocolic flexors. Alright, so the mass movements, they're going to occur especially in the transverse colon, which is the one that goes across, and often within an hour after breakfast. Now, stretching of the rectum, it stimulates the 2 defecation flexors, which are often felt soon after a meal. Now, the intrinsic defecation flexor, it operates entirely within the myenteric nerve plexus. And the stretch signals, they travel through the plexus to the muscularis of the descending and the sigmoid column and the rectum. Now, a peristaltic wave is activated that drives feces downward and relaxes the internal anal sphincter. Let me get a pen here. Okay, so when we deal with the intrinsic, we're dealing with signals that are going to travel from the plexus of the muscularis of the descending and the sigmoid column and the rectum. So we're looking at the descending, the sigmoid, and then the rectum. This area where we're dealing with the intrinsic defecation reflex. Now we talk about the parasympathetic defecation reflex. It's a spinal reflex. So the stretch signals are transmitted to the spinal cord. We have motor signals. Motor signals return by way of the pelvic nerves to intensify the peristalsis in the descending and the sigmoid colon and rectum, and to relax the internal and the internal anal sphincters. Now these reflexes are involuntary. Let me go back here. These reflexes, they're involuntary and are the sole means of controlling defecation in infants and people with trisectin spinal cord injuries. Now the external anal sphincter, like the external urethral sphincter, is under voluntary control, which I mentioned earlier. And defecation normally occurs only when this sphincter is voluntarily relaxed. Now, defecation is also aided by the valsalva maneuver. Now, defecation urge is suppressed. Contractions cease in a few minutes, and the rectum starts to relax. The reflexes occur again after a few hours later. So here we have the neurocontrol of this defecation, where we see that we have the parasympathetic nerve fibers, motor fibers. They're being signaled to relax the internal anal sphincter. Here we have it here, the internal anal sphincter. And to contract the rectum. So the rectum is being contracted, and we have the relaxation of our internal anal sphincter. And here we have the spinal reflexes or reflex are relaxed. It starts to relax that internal anal sphincter. Here in number 3. And in number 4, which we have here, we're coming from the voluntary motor signals. You have these impulses from the brain that are going to prevent untimely defecation. So when you're not ready to use the restroom or to defecate, you're here controlling it. And what you're going to do, you're going to be contracting the external anal sphincters just to hold off the defecation. So defecation is only going to occur if this sphincter is relaxed. So it will sit here for a while until you're ready to defecate, or until you have relaxed the external anal sphincters. So everything has to be relaxed, and both sphincters need to be relaxed in order for defecation to occur. So here's an overview. I got it from another textbook. An overview of the function of the gastrointestinal organs. You have your mouth and the accessory organs. You have the ingestion, propulsion, mechanism, mechanical digestion, chemical digestion, propulsion that occurs. You have also mechanical digestion and propulsion being within the pharynx and esophagus, as well as the chemical digestion within the stomach. We have the digestion of the proteins. And what did I say in the mouth? Which one is digested in the stomach, in the mouth first, in the oral cavity? Is it starch, fat, or carbohydrates? It's our carbohydrates. Our carbohydrates are digested here. Alright, then we have our small intestine and associated accessory organs that deliver the gallbladder and the pancreas. And here you have the various things that are occurring, the major functions of this particular area. Then we have our large intestines, where we deal with, we have the chemical digestion, where we have food residue that remains in the area. We have the absorption, Some of them remain water, electrolytes, and the propulsion, which is dealing with the propelling of feces toward the rectum. And then we have the defecation, which is the final stage where the feces are eliminated from the body. Alright, so that was dealing with those areas. Now let's get on to the chemical digestion and absorption. So we're going to be looking at the major classes of the nutrients being digested, as well as the various enzymes that are involved in it. So let's move on. I kind of switched it up in your textbook. I wanted to stick with the whole process and then come back to what happens when you eat these particular foods. So let me deal with starch. Now, most of the digestible carbohydrate is starch. Cellulose is indigestible. Now, starch is digested first to oligosaccharides up to 8 glucose units long. Then into disaccharide maltose, and then finally into sucrose. So if you guys remember from chemistry, where we have our 8. Let's see if I can get this drawn here for you quickly, as you can see. So here with our starch, we'll say we have 8 little molecules. This is our oligosaccharides, and then they're broken down again into our disaccharides, which should just be 2 glucose molecules, and then into our glucose molecule. So that's how things are broken down, and this is what's being absorbed into the intestines, absorbed by the intestines, which is this glucose. It has, remember, things have to be broken down into its simplest form in order for it to be absorbed and utilized. Okay, let's make sure I didn't skip anything. Okay, here we go. So now beginning in the mouth, we have the salivary amylase, which hydrolyzes starch into these oligosaccharides. So this enzyme basically is going to break down the starch into the oligosaccharides. Now the enzyme, this salivary amylase, it functions best at a pH of 6.8 to 7, typically by the mouth. Now it is denatured upon contact with the stomach acid, but if it is protected in the middle of the food mass, it can continue to digest for as long as 1 to 2 hours. Now, about 50% of the dietary starch is digested before it reaches the small intestine. Now, where pancreatic amylase continues, here you have the pancreatic amylase, which comes from the pancreas, the enzyme, where this continues is pancreatic amylase continues the process. So once that starch enters in, we have the breakdown of the starch to the oligodendrocytes. Then of course we have 50% of the starch is broken down by the pancreatic enzyme when it reaches into the small intestines. When the small intestine starch is entirely converted to the oligodendrocytes and maltase within 10 minutes. Now digestion is completed as chyme contacts these brush borders and is exposed to the enzymes. So these are our brush borders here we've just talked about in a couple of videos. Now 2 enzymes. Okay, so when the chyme makes contact with the brush borders, we have these enzymes here that are that the chyme is going to come into contact with, and that's the maltase, the dextrinase, and the gluco uh, amylase. Sorry, glucoamylase. Now, two of the enzymes, the very last two, the dextrinase and the glucoamylase, they're going to hydrolyze the oligosaccharides that are 3 or more residues long. So here you have the oligosaccharides that are coming along. They're going to brush against the brush border, and the dextrinase, the dextrinase and the glucoamylase, they're going to hydrolyze it. Now the third enzyme, the maltase, they're going to hydrolyze the maltose, and going to break the maltose down to glucose. Alright, so the major dietary disaccharides are sucrose and lactose. I'm going to move on to the next slide so you can follow along with me. So here I am. They are digested by the brush border enzymes sucrose and sucrase and lactase, and the resulting monosaccharides are immediately absorbed. Now, sucrose consists of glucose plus fructose. Now, lactose consists of glucose plus galactose. So let me see if I can pull up a page for you guys. This time. Okay. All right. So the two dietary disaccharides. My disaccharides are two glucose molecules. They are sucrose that's an S, sorry. Fix that. Sucrose and our lactose. Now, the sucrose consists of glucose plus fructose. And our lactose, two molecules, it is glucose plus galactose. Hopefully you guys can make out what I'm trying to write here. Now, most of the world population, this lactose here becomes indigestible after about age 4. Okay, so just give you an idea of what we're dealing with. Alright, so the plasma membrane of the protective cells, it contains transport proteins that are going to absorb these monosaccharides. Remember, we talked about these monosaccharides. It's broken down to its simplest form now. Now, about 80% of the absorbed sugar is glucose. Now, glucose is taken up by a sodium glucose transport protein, or SGLT, like that of the kidney tuberose. Now, the glucose is then transported out of the base of the cell and into the extracellular fluid. Now, sugar entering that extracellular fluid, it increases its osmolarity, and water is drawn from the lumen of the intestines through the low leaking tight junctions. Now, water carries more glucose than other nutrients by a solvent drag, as much as 2 or 3 times as much as glucose is carried by the SGLT. Now, the SGLT also absorbs galactose. Now, fructose, in contrast, is absorbed by facilitated diffusion, usually using a separate carrier that does not depend on sodium. Now, inside the epithelial cell, most fructose is converted to glucose. So you have glucose, galactose, and the remaining fructose are transported out of the base of the cell by facilitated diffusion. And then they're absorbed by the blood capillaries. Now, the monosaccharides are then delivered to the liver by the hepatic portal system. Now, we do have some people that are lactose intolerant. Those are people without that lactase. Now, lactose passes undigested into the large intestines, so that lactose is not going to be broken down by that lactase, because they don't have that lactase. So it's going to increase the osmolarity of the intestinal contents, and it's going to cause the water retention in the colon and of course diarrhea. Now, gas production, we do have gas production by the bacterial fermentation of the lactose. So don't forget, when we're dealing with lactose intolerant, we're dealing with individuals that are missing that enzyme lactase. Right here, lactase, because this lactase is there to break down this lactose. So it's going to go into the intestines without being broken down. Now, it occurs in many people. Approximately 15% of American whites suffer from lactose intolerance. 80% in African Americans, 70% Mediterraneans, and nearly all of the Asian descent. Some individuals that consume yogurt, cheese, or drink milk. Some people can consume those because the bacteria that's in the cheese and the yogurt can actually break down that lactose. So some people can eat yogurt and cheese. Some people can't eat cheese, but I know a lot of people can eat yogurt and not have any problems with lactose intolerance. So a lot of people who do have lactose intolerance, they resort to different types of milk, maybe silk milk or almond milk, as a substitute from dairy milk, which is your 2%, 1%, and your whole milk. Alright, so let us take a look at our proteins. Now, our amino acids, they come from three protein sources. We have our dietary proteins, our digestive enzymes digested by each other, and the slothed epithelial cells. Now, endogenous amino acids from the last two sources total about 30 grams per day. So we're talking about these two sources right here. Okay, and that's compared to 40 to 60 grams per day from our food, which is here. So these last two make up about 30 grams per day, and the very first one makes up the 44 to 60 grams per day. Now, enzymes that digest proteins are called peptidase, or let's call it peptidases, peptidases. Okay. Now, in the stomach, the pepsin, they're going to hydrolyze the peptide bond between the tyrosine, and the phenylalanine, and they're going to break it down 10%, breaking 10 to 15% of the dietary protein into shorter polypeptides and some free amino acids. Now, pepsin has an optimal pH of 1.5 to 3.5 and is inactivated when it passes into the duodenum, and then it's going to mix with the alkaline pancreatic juices, which have a pH of 8. Now, in the small intestine, the trypsin and the trimotrypsin from the pancreatic enzymes, they continue hydrolysis. Now, the shorter oligopeptides are then taken apart by 3 more enzymes. And what are those enzymes? We have carboxypeptidase, aminopeptidase, and dipeptidase. Now, the first one, the carboxypeptidase, that's coming from the pancreas. So it's a pancreatic secretion. Not excretion, secretion. And they're the ones that are going to remove the amino acids from the carboxyl group. Okay, this is considered to be the carboxyl group. And this is usually the ones that you find at the end of a chain. Okay. Now the second one, the aminopeptidase, is one of the enzymes you'll find along the brush borders, and they're ones that are going to removes them from the amino, I can't remember, the NH2 end. The last one, the dipeptidase, also a brush border enzyme. It splits the dipeptides into middle. In the middle, it splits the dipeptides in the middle, and then it's going to release the last 2 amino acids. So here we have an example of the proteins being broken down. Here we have our trypsin, which you see here, and then we have our trimotrypsin. And the green arrows is our carboxypeptidase. You see how they're removing that carboxyl group. It's removing that carboxyl group from the end of the polypeptides. Okay, so here's the polypeptide, and you see how it's going through hydrolysis, breaking down, and we have the carboxyl group that will eventually be removed. It just removes 1 amino acid. So here we have approximately 5. 1, 2, 3, 4, 5. Here's your amino acids, the blue circles or the blue beads. You have the carboxypeptidase that comes in and removes just 1 amino acid. So here we have 1, 2, 3, 4, 5, 6. The enzyme comes in, removes an amino acid, leaving us with 1, 2, 3, 4, 5. It removes again, and we're left with 1, 2, 3, 4. Removes another amino acid. Here we are left with 3. So it removes it from the end from this end, the carboxyl end, not this end here, not from the end, not from the head, but from the tail. Okay, always from the end, it's going to remove it from this particular end. Okay, so in this diagram here, your figure 25.29, you see those 3 enzymes coming into play, and breaking and removing various amino acids. So I showed you the very first one, the carboxypeptidase. So but when you look at the second enzyme, which is the aminopeptidase, it removes it from the amine end. So here's your amine end, here's your carboxyl end. So it's going to remove an amino acid from the amino end. Then your dipeptidase will just split it down the middle. Okay, so that kind of makes sense what I was referring to, and also what was in your notes a couple slides back. Okay. Okay, so the amino acid absorption is similar to that of the monosaccharides. You're going to have several sodium-dependent amino acid co-transporters. So those ones are going to help carry these amino acids. Carry them, different classes of amino acids. They're going to carry them around the membrane. Now the dipeptides and the tripeptides, they may also be absorbed but are hydrolyzed within the epithelial cells. Now, at the basal surfaces, amino acids, they leave the cell by facilitated diffusion, and they enter the capillaries and are carried away in the hepatic portal circulation, which we see right here with the blood vessels. We see these amines are going to be carried away. So they go through facilitated diffusion, they're going to enter into the capillaries, and then are carried away by the hepatic portal circulation. Now the absorptive cells of infants can take up intact proteins by pinocytosis, and then release them into the blood by exocytosis. Now this allows the IgA, circle that here, IgA, where's the IgA from breast milk to confer passive immunity. Now intact proteins, they're going to enter the infant's blood. Intact proteins entering the infant's blood may be treated as foreign antigens and sometimes trigger food allergies. Now the ability of the intestines to pinocytose protein, it declines but never completely ceases. Now the hydrophobic quality of lipids makes their digestion and the absorption more complicated than that of the carbohydrates and the proteins. Fats are digested by enzymes called lipases or lipase. Please remember that. You will see that word again. Now lingual lipase, secreted by the intrinsic salivary glands of the tongue, they digest a small amount of fat while food is still in the mouth, but becomes more active in the acidic pH of the stomach. Now gastric lipase, it makes a much larger contribution to pre-duodenal fat. So that's the fat that's right before entering the duodenum portion of the small intestine. So that's right before it gets into the duodenum. So you know it's still in that pyloric region of the stomach. And the gastric lipase makes a larger contribution to help breaking down the fat. Okay, now most fat digestion occurs in the small intestines through the action of the pancreatic lipase. Now fat in chyme is in large globules as it enters into that duodenum. Okay, now it is broken up into smaller emulsification droplets by compounds of the bile and the lecithin and the bile salts. Now intestinal segmentation also breaks the fat up into droplets as small as 1 micrometer in diameter and a coating of this lecithin and the bile salts keep it from breaking up, exposing its surface to the enzymatic action. Now there's enough pancreatic lipase in the small intestines after a meal to digest the average daily fat intake in 1 to 2 minutes. Now lipase, it removes the first and third fatty acids of the triglycerides and leaves the middle one, producing 2 free fatty acids and a monoglyceride. So here we have in this diagram talking about emulsification. So we have the lecithin and we have our bile. We see it has a hydrophilic and a hydrophilic regions of the bile. So when this fat droplet or globule enters the intestinal segmentation, what's going to do, it breaks the fat into these droplets, okay, and it's going to coat it with the lecithin and with the bile. So now this fat goblet is broken down and then it's coated with these lecithin and the bile. Now absorption of fatty acids, monoglycerides, and other lipids depends on minute droplets in the bile called micelles. These micelles consist of 20 to 40 bile acid molecules aggregated with hydrophilic side groups facing upward. And the hydrophobic steroid rings are going to face inward. Now the bile salts, the bile phospholipids, and the cholesterol diffuse into the center of the micelle to form its core. Now, lipids, as we see, are made in the liver. Okay, so the micelles, they're going to pass down the bile duct into the duodenum, where they absorb fat-soluble vitamins, more cholesterol, and the products of fat digestion. Now, their hydrophilic surfaces allow them to remain suspended in water. They transport lipids to the surface of the intestinal absorptive cells, where the lipids leave the micelles and diffuse into the cells. The micelles are then reused. Now, without these micelles, the small intestine absorbs only about 40% to 50% of the dietary fat and almost no cholesterol. All right, so here we have this example of our fat hydrolysis, and we have our lipid uptake by these micelles. So here we have our monoglyceride and we have our fatty acid. Okay, so this is referring to what I was talking about earlier, where we have the pancreatic lipase coming in and removing the first and the third phospholipids. So we talked about our bile, our dietary lipids, and how it's surrounded by lecithin and the bile acid. Then we have the pancreatic lipase that comes in and starts breaking it up. It'll take it apart from the first and the third, and this leave you with 2 free fatty acids and 1 monoglyceride. This monoglyceride here now and other lipids is going to depend on these little droplets of bile called the micelles. Okay. Now these micelles, which I told you about, they have consist of what, 20 to 40 bile acid molecules. Now the bile, phospholipids, and the cholesterol, they diffuse into the center of the micelle to form its core. So in here, you're going to have the bile, phospholipids, and the cholesterol. All these make up the core. Okay? Then the micelle is going to pass down the bile duct into the duodenum, where they absorb the fat-soluble vitamins, more cholesterol, and the products of the fat digestion. Okay. Alright, so let's continue with our lipids. Alright, so with the enterocytes, the free fatty acids and the monoglycerides are transported into the smooth endoplasmic reticulum, the smooth ER, and they're resynthesized into triglycerides. So here we see in this particular diagram here, made into triglycerides. Now the Golgi apparatus, it combines these triglycerides with a small amount of cholesterol and coats the complex with a film of phospholipids and proteins to form our droplets called chylomicrons. Here we go, right here. Okay. Now it packs these into these vessels that are going to migrate to the basal membrane of the cell and release their contents into the core of the villi, or the villus. Sorry. Now the chylomicrons are too large to penetrate this epithelium of the blood capillaries, and are instead taken up by more lactose in the lymphs. Now the fatty intestinal lymph eventually flows through the cisterna chyli to the thoracic duct and then into that bloodstream at the left subclavicular vein. Here we go. This diagram here for you guys to look at. Alright, so while they're picked up by the Golgi apparatus, sorry, okay, so after they're packaged here, they're put into these vesicles, then they're going to be taken to the basal surface of the cell. Then they're going to release their contents into the core of the villus. Now the chylocrones, as mentioned, they're too large to penetrate the blood vessels endothelium, sorry. So instead of being taken up by the lactose into the lymphs, so they're taken up by the lymphs. I'll get this working here. So they're taken up in here in the lymphs, the porous lactose in the lymph nodes, or in the lymphs. Alright, so here is a summary of what is occurring when carbohydrates, proteins, and fats are digested. Here it is on the table when we consume it. The starch, when it comes to carbohydrates, you have the salivary amylase that breaks it down into maltose and oligosaccharides, as well as lactose and sugar. This is broken down in the mouth. When it reaches the stomach, it's still broken down. When it gets in the small intestines, we have the pancreatic amylase that is secreted by the pancreas, and it breaks down the these two, the oligodendrocytes, no, oh my goodness, oh my word, the oleosaccharides into maltose. So we have maltose coming all the way down, and now they're ready to enter into the villi, the villus, and the lining of the epithelium of the small intestines. When it reaches here, it's converted into glucose by the maltase. When glucose is here, it reaches into the blood capillaries, or the blood capillaries, where it is circulated and utilized. The same, similar with the lactose and the sucrose. We have the lactose and the sucrose lactase that breaks down the sucrose and the lactase, giving us fructose and galactose. You have the fructose that come over, that is converted to the glucose, and we have the galactose that's broken down. That enters into the blood capillaries. So you can see by the diagram, it's very self-explanatory as you see where the enzymes come into play, either in this mouth, in the oral cavity, in the small intestines, for each group: the carbohydrates, the proteins, and the fats. Okay, so that's everything I was just referring to. Here's your beautiful diagram to help you with understanding what is going on with nutrient digestion. Alright, so when we look at the nucleic acids, they are present in only small quantities compared to other dietary monosaccharides. Now the nuclease, which are the RNA and the DNA of the pancreatic juice, they hydrolyze these nucleotides. Now the nucleosides and the phosphatase of the brush borders then break these into phosphate ions, ribose or deoxyribose, and our nucleic bases. Now these are transported by the membrane carriers, and then they're going to enter into the bloodstream. So let me do another diagram. We have our RNA, and we have our DNA. That is a D. Oh, okay, let me see if I can erase that. This is my D. Didn't look any better. And there's my DNA. So when we have these two nucleases, they're gonna hydrolyze the nucleotides. So we have these enzymes called, they're in your textbook, also on the previous slide, the nucleosidase, that's a D, ASES, and the phosphatase. They help break down the RNA and DNA into ribose and deoxy, that's the X, into deoxyribose. Okay. Alright, so let's look at vitamins. Vitamins are absorbed unchanged, which is good. Now the fat-soluble vitamins A, D, E, and K are absorbed with other lipids. If they are ingested without fat-containing food, they are not absorbed at all. So those who are trying to consume potassium or vitamin K, A, D, and E, you need to have some type of fat within your food. Good fat, sorry, dietary good fats, to help with absorption of these. They're just going to be passed straight out as waste and into your feces. So there's nothing wrong with consuming some fats, because it does help with the absorption of the A, D, E, and K. Alright, that's why they're called fat-soluble vitamins. Now, the water-soluble vitamins, our B complex and our vitamin C, are absorbed by simple diffusion, with the exception of our B12. These large molecules can only be absorbed if it binds to intrinsic factors from the stomach. Now, the B12, that intrinsic factor complex on them, binds to factors on cells of the distal ileum, where it's taken up by the receptor mediated endocytosis. Sorry, kind of skipped ahead. Alright, so when we look at minerals, also our electrolytes. Everybody knows about electrolytes, especially those who drink Gatorade, Powerade, trying to get the electrolytes back into your system. Those are absorbed along the entire length of the small intestines. We have the sodium ions are co-transported with sugars and amino acids. The chloride ions are actively transported in the distal ileum by a group that exchanges them from bicarbonate ions. Now, the reverse of the exchange that takes place in the stomach. The potassium ions are absorbed by simple diffusion. The potassium concentration of chyme, it raises as water is absorbed, creating a gradient that favors potassium absorption. Now, in diarrhea, when water reabsorption is hindered, potassium absorption is hindered also. This can also lead to hypocalcemia, which is low potassium. Alright, or low calcium. Alright, so in iron and calcium, these are usually found in that they are absorbed in proportion to the body's needs, whereas other minerals are absorbed at a constant rate. So if your body doesn't need the iron or the calcium, it's not going to absorb it. Now, iron absorption is stimulated by the liver hormone hepcidin. Now, the absorption cells, they're going to bind the ferrous ions, the F2 ions, but are unable to absorb the Fe3. Now, the stomach acid, it reduces more Fe3 ions to Fe2 ions. Now, the Fe2 ions are transported to the basal surface of the cell and are taken up by the extracellular protein transferrin. Now, the transferrin iron complex, it diffuses in the blood. Let me go back. Skipped again. I'm sorry. This complex, the transferrin iron complex, it diffuses into the blood and is carried to the bone marrow for hemoglobin synthesis, muscle tissue for myoglobin synthesis, and then to the liver for storage. So it's going to 3 different places: the bone marrow, the muscle tissue, and the liver. The bone marrow for hemoglobin synthesis, the muscle tissue for myoglobin synthesis, and then the liver for storage. Now we look at calcium. Calcium is absorbed throughout the intestine, some by active transport in the duodenum and more for diffusion between the epithelial cells in the duodenum and the ileum. Now in the duodenum, the calcium ions are going to enter the absorptive cells through calcium channels in the apical cell membrane. Now inside the cell, it binds to a protein called calbindin. And it's going to keeping the intracellular concentration low, maintaining a gradient favorable for uptake. Now free calcium in the cytoplasm is pumped out the basal side of, pumped out of the basal side by this calcium ATPase as well as the sodium calcium antiport. And from there to the blood capillaries. Alright, so let's take a look at what I was talking about with our calcium. So we're dealing with just calcium ions. Alright, so here we have our, we'll just draw a little something with our duodenum. And the duodenum, we'll just have it like this. Here's our duodenum. So in the duodenum, the calcium is going to enter the absorption cells through these channels. So here we have these channels. This is our, let's say our duodenum. Oops. Our duodenum. And these calcium, they're going to enter into the intestine, the duodenum, through these channels. And these channels is what you have in the apical membrane. So this is the this is duodenum, but here is your membrane here. Okay. Now, when it's inside the cell, it's gonna bind. Let me make a little cell here. Alright. So inside the cell, we're gonna have it binding to this protein called, I'll just put P for protein. But that protein is called binding. So that's my P in here. Let's see if I can just fix that up just a little bit. Go back to my pen. Alright. Alright, so here is my little protein. So when the calcium comes in, change it back to blue for calcium. When the calcium enters the cell, it's going to bind to that protein. And it's going to keep this intracellular, remember this is intracellular, this is extracellular, so it's going to keep this concentration low, which is what we want. It's going to maintain the gradient for any type of uptake. Okay. Now we do have these free calciums in the cytoplasm, and they're pumped out of the basal side. So now we have calcium being pumped out. And they're pumped out through these various channels. And this is out of this is called a sodium calcium antiport. Okay, and what helps them leave is this enzyme. Get my black one over here. Helps them leave is this calcium ATPase. So I'll just put calcium / ATPase. That is a T. Okay, there we go. And when they leave this port, this antiport, they're going to enter into our, I think it's in red, into our blood capillaries. So here are my blood. Okay. Alright, so let's get on to our parathyroid hormone. That is secreted in response to any drop in our calcium levels. So if the calcium levels are too low, the parathyroid is going to be secreted by the parathyroid hormone is going to be secreted by the parathyroid. And with that drop, it's going to stimulate the kidneys to synthesize that vitamin D. Now, these effects this affects the absorptive cells of the duodenum in three ways. The number of calcium channels are going to increase. The number of calbinin in the cytoplasm is going to increase. And the number of calcium ATPase pumps in the basal membrane are going to increase. Now, the vitamin D has no effect on the paracellular diffusion of calcium in the duodenum and the ileum, where most of the calcium is absorbed. So all this is happening in the duodenum. Now, the digestive system is one of the systems involved in water balance. The digestive tract receives about 9 liters of water per day from all sources. Now, about 8 liters of this is absorbed by the small intestines and 0.8 liters by the large intestines, leaving 0.2 liters voided in the daily defecation output. So let's get my little diagram here again that we use. So that's fine. So out of the 9, so let's do this right here. So we have a total of 9, correct? 9 liters of water per day. So you see how it's very important that we drink water every day, because it is essential with the breaking down of breaking down of the foods, as well as helping with defecation. So a total of 9 liters per day. I can't get my day written in here, but you see it right here. And we have, okay, there you go. We have, of those 9, we have 8 liters, which is absorbed by the, I'll do abbreviations, by the small intestines. We have 0.8, let me point this off, 0.8, which is absorbed by the large intestines, and then we have this 0.2, which is voided out, basically through defecation. Okay, so basically if you're constipated or you have hard stool, is what it's called, you have hard stool, you're not consuming the right quantity of water, because your small intestines need most of it. So your small intestines is taking up all of it if you're not consuming your 9, because it has to have this amount, a certain amount, in order for you to help with the digestion of the food. Alright, let's move on to the next page. If we can get it off. Okay, here we go. Alright, so water is absorbed by osmosis following the absorption of salts and organic nutrients that pass into the extracellular fluid. Now, diarrhea occurs when the large intestines reabsorb too little water. This may occur when the intestines is irritated by bacteria and feces pass through too quickly. Now, it may also result from abnormally high concentrations of solutes such as lactose that poses osmotic absorption of water. And then we have our constipation. Constipation occurs when fecal movement is slow. Too much water is reabsorbed and the feces become hardened. It can result from lack of dietary fiber, lack of exercise, emotional upset, or long-term laxative abuse. So we are at the end of our chapter 5. It is a lot of information, very detailed. Please, if you have any questions, make sure you post it on Blackboard. Don't email me, please. Just post it on Blackboard so everybody can see the question that you have and see my response or anybody else's response. Okay? Thank you so much, and good luck on your tests coming up.